Here, you're going to learn all about mitosis. In multicellular organisms, mitosis occurs within the cells to help the organism grow and develop. However, in unicellular organisms, when mitosis occurs, more of these unicellular organisms are created. During its life, the cell goes through a cell cycle. It starts off at the G1 phase, their cell growth and differentiation. Then it goes to the S phase, which is DNA replication. Finally, it goes to the G2, which is another growth stage. All this occurs during interphase of the cell cycle. Then the cell goes through mitosis, which consists of prophase, metaphase, anaphase, and telophase. However, after the G1 stage, the cell may go into G0, or non-dividing state, and on certain circumstances, it may return to the cell cycle. Most of the cell's life is spent in interphase, which consists of three separate stages: G1, the first growth stage; S, the stage responsible for DNA synthesis; and G2, the second, smaller growth stage. The DNA is left in a loose state called chromatin, which is replicated during the S phase, and during this replication, sister chromatids are formed and held together at a centromere. The cell, during interphase, is just doing its job and doing what a cell does. During early prophase, the DNA condenses into chromosomes. The nuclear membrane begins to break down and becomes less visible. The centrosomes move to opposite poles and the spindle fibers are formed. During late prophase, the DNA is fully condensed into chromosomes. The nuclear membrane is gone and not visible at all, and the chromosomes attach to the spindle fibers. In metaphase, the spindle fibers position chromosomes along the center of the cell. During anaphase, the spindle fibers shorten, pulling the sister chromatids apart to opposite poles of the cell. During telophase, the sister chromatids reach the poles, and the events of prophase reverse. The DNA decondenses, the spindle fibers disappear, and the nuclear membrane reforms. Additionally, cytokinesis begins, where a ring of protein fibers pinches the cell in two until the daughter cells can separate. However, in plant cells, cytokinesis is different. Carbohydrate-filled vesicles bud off of the Golgi apparatus and move to the center of the cell. These vesicles fuse to form a new cell wall and plasma membrane between the daughter cells until there's complete separation of the cells. Throughout the cell cycle, the cell goes through various checkpoints, one between the G1 and S phase, another between the G2 phase and the beginning of mitosis, and a third and final checkpoint between metaphase and anaphase. If the cell does not meet all the checkpoint requirements, the cell cycle is arrested, and the problem can either be fixed or the cell can perform apoptosis, which is essentially suicide for the cell. One major checkpoint is the R checkpoint, which occurs between the G1 and S phase. If the cell passes this checkpoint, it usually finishes the entire cell cycle. And these checkpoints are controlled by the activity of CDK proteins, which are always present in the cell. And one major control protein in the cell is PRB. During a normal DNA sequence, during the G1 through S checkpoint, the R checkpoint, growth factors and the receptor proteins fuel cyclin synthesis with the use of CDK, which eventually leads to the replication of DNA. However, if there is DNA damage, p53 helps block RB phosphorylation, which stops DNA replication, preventing the replication of the damaged DNA. If there is a mutated growth factor receptor gene, the mutated receptor is always on, so when it goes through the phases with cyclin synthesis and CDK to replicate DNA, the replication is uncontrolled. If there is a mutated cyclin gene in this process, instead of the receptor being always on, cyclin synthesis is always on, which in turn also leads to uncontrolled DNA replication. If there is a mutated RB gene in this process, the mutated RB does not require phosphorylation. Because it does not require phosphorylation, the DNA replication becomes uncontrolled. If there is a mutated p53 gene and DNA damage, the mutated p53 cannot block RB phosphorylation. Thus, DNA replication occurs and is uncontrolled. All of these mutations are caused by oncogenes and speed up the cell cycle, and activation mutations can lead to cancer. Tumor suppressor genes can slow down the cell cycle, and deactivation mutations can also lead to cancer. That's all you need to know about mitosis. Thank you for watching.